Hey there, future biologists. Welcome back to another exciting journey into the microscopic world that shapes our lives. If you've ever wondered how scientists copy and manipulate DNA, you're about to unlock the secrets of cloning vectors. These tiny tools are the unsung heroes behind some of the biggest breakthroughs in genetics and biotechnology. Let's dive right in and see how they work. So have you heard of plasmids and bacteriophages? These are the original DNA carriers that nature designed long before humans ever thought about genetic engineering. These tiny DNA molecules are nature's own copy machines inside bacterial cells. They exist separately from the main bacterial chromosome, quietly doing their own thing. Unlike the main chromosome, plasmids and bacteriophages replicate on their own, almost like freeloaders with a VIP pass. They don't need permission from the cell's main DNA to start making copies of themselves. Now, here's something wild. Bacteriophages, which are basically viruses that infect bacteria, can have super high copy numbers. When they invade a bacterial cell, they hijack the cell's machinery to make loads of copies of their own DNA. Imagine dozens, even hundreds of copies of their DNA packed inside one cell. It's like a tiny factory churning out identical blueprints at lightning speed. Plasmids, on the other hand, can range anywhere from just a couple to 100 copies per cell. Some are low copy, quietly hanging out in small numbers, while others are high copy, multiplying like crazy. And yes, sometimes those numbers can climb even higher under the right conditions. Scientists can tweak the environment or the plasmid itself to boost those numbers, making plasmids even more useful for research. But why does this matter? Why are scientists so obsessed with these tiny DNA circles and viral invaders? Well, if you can attach or link a piece of foreign DNA, let's call it alien DNA, to a plasmid or bacteriophage, you can use their natural ability to make tons of copies. This is the foundation of DNA cloning, a technique that has revolutionized biology. That means multiplying your DNA of interest until you have enough for experiments, research, or even making medicines. Imagine being able to produce enough insulin for millions of people, all thanks to these tiny vectors. Now, here's where the magic of modern science comes in. Scientists have taken what nature gave us and improved it, creating vectors that are more efficient, safer, and easier to use. The vectors we use today aren't just borrowed from nature, they're engineered for efficiency. They're like the latest model of a smartphone, packed with features that make genetic engineering faster and more reliable. These cloning vectors are designed so it's super easy to insert your foreign DNA. Special sites called restriction sites act like molecular scissors, letting scientists cut and paste DNA with precision. Plus, they come with built-in tricks to help you spot which bacteria actually took up your DNA, called selection markers. These markers might make bacteria glow, change color, or survive in the presence of antibiotics. So, in simple terms, cloning vectors are like customizable delivery trucks. They pick up your DNA cargo, deliver it into bacteria, and then make hundreds or thousands of copies, all without needing the main DNA's permission. This process is the backbone of genetic engineering, allowing us to study genes, produce proteins, and even create genetically modified organisms for agriculture and medicine. To recap, plasmids and bacteriophages are the workhorses of molecular biology because of their ability to self-replicate. Without them, many of the advances in modern science simply wouldn't be possible. By linking foreign DNA to these vectors, we can harness their natural machinery to multiply DNA for all kinds of purposes, from genetic research to biotechnology, agriculture, and even environmental science. If you found this video helpful, hit like and don't forget to subscribe for more insert-based biology explanations. Your support helps us bring more science to curious minds like yours. Got a topic you're curious about? Drop it in the comments and I'll cover it next. Whether it's about genetics, cell biology, or any other science mystery, I'm here to help you explore. Thanks for watching, and keep exploring the wonders of biology. Remember, every big discovery starts with a tiny question, so stay curious and keep asking why.